Thank you all to be here for the seminar of Diatchenko professor. I leave the floor to Professor Fumagalli for the presentation of our speaker. Thank you.
Thank you. So I'm Roberto Fumagalli. I'm a full professor of anesthesia and intensive care at Bicocca University. And today I have the privilege and the honor to introduce Professor Luda Diatchenko. Professor Luda Diatchenko is one of the most important researchers in the field of chronic pain. Basically, she is performing studies basically on the experimental model about how pain becomes chronic. That's a quite important issue because let's say, just to make an example, 30% of the women who have undergone to breast surgery, after 1 year, they still have pain. And this happens for many other situations. So chronic pain is a really important issue. Moreover, there is not just one specialty who is taking care of chronic pain. Many specialties are dealing with it, but without a great success. So who is Luda Diatchenko? Luda Diatchenko, she is, I have to read because it is very, very huge. She is actually full professor and research chair in human pain genetics, fellow of the Royal Society of Canada, and she is working at the McGill University. She has a very huge lab over there, and a lot of persons is cooperating with her. So let me stop here and let me introduce Luda Diatchenko, and she will tell us about very exciting and new findings in chronic pain. Luda?
Professor Fumagalli, thank you for generous introduction. Thank you everybody who come and those who came also online. So today I will talk about how we develop chronic pain, or what I found more useful to think, how we actually resolve pain. And I will start with this paper, which we published in Science Translational Medicine 2 years ago. And this paper created a lot of discussion. It was #1 in all metrics for a while for Science Translational Medicine. And so what I would like to tell you, so like, what is the controversy about our finding? And I think I also will convince you that there is no actually controversy. It's just the next step of our understanding. So I'm a human geneticist. Yeah, so it's very important. Human geneticist. I study pain, but I always start from human samples, because as many other field, because I don't think we understand pathophysiology of chronic pain, any animal model does not exactly represent human pain. So I would rather start from human samples. And so in this case, we started with cohort, which actually has been collected in Parma by Dr. Massimo Allegri. And the cohort was of the people with acute back pain. So we had people samples, blood, RNA at the acute stage in 3 months later. And after 3 months, So some people resolve pain, we call them resolvers, and some people still have pain, we call them persisters. So then what we did is we, means Mark Parisian here, our genius bioinformatician in the group. So we did full analysis of the RNA in the blood of these people. Okay, so this is so-called volcano plot. And so we compare all transcriptomics, how it's changed in the people who resolve pain from acute to 3 months. So if you're not used to look at the volcano plot, okay, it's very easy. I can orient you. So each dot is the gene. And so here is a log p value of how significant is its change in the expression of the gene. The blue line is FDR correction for full transcriptomics. So basically all pink dots are genes which significantly change expression after correction for all transcriptomics, right? So you can see this number is how many genes, and there is an upregulated and downregulated gene. There is thousands of genes, thousands. It's basically half of the genome change expression in the people who resolve pain. And so what's happening in the blood people who don't resolve pain? You see, it's absolutely nothing. I mean, you don't often see black and white in biology, right? So, but we get lucky with these samples. There is absolutely nothing happening in the people who don't resolve pain. So, and I was personally kind of confused with the samples, and I asked myself, like, what confused me? And what confused me, I realized, is because we always think about how we develop pain, right? So we always think about, or me anyway, and many on the field, is that kind of there is some active pathological process happen, and we need to find this gene, and it will be our drug target, which we will need to target. Well, this is kind of show that there is difference. So it's the way how we resolve pain is an active process, and pain chronification is the absence of this adaptive activity in the immune system. Or at least it's associated with this. Yeah, so we don't know what's primary, but at least there is a huge association. And it doesn't seem it's one gene. It's a river. Yeah, it's a river going down. It's like I'm from originally from Kazakhstan, so I live in the mountains. So I imagine this, in the spring, this water going down. Yeah, like you can stop it. It's, if you put any even big block in the middle, it's just will washed away. This was my first kind of conclusion. And then we thought how to analyze this data, because again, it's not one gene. Our usual things, we publish paper, we say, okay, this is the most important gene, and we study it. So, okay, we decided to do pathway analysis. I mean, it's also standard. It was not a huge breakthrough to do pathway analysis. But we did pathway analysis, and the pathway analysis was most interesting and telling when we compare pathway analysis in the acute stage. So what differentiates people who will resolve and will not resolve pain at the acute stage? And so the most significant pathway was biologically specific, because it was a lot of, I don't know, biological process, signals, cell signaling. But in terms of specific biological process, it was inflammatory pathway, which wasn't surprising because you expect to have some inflammatory activity in the people with acute low back pain. What's surprising was the direction, because direction was such as that it's people who will resolve pain have a lot of, so this is again a logarithm of FDR. So people who resolve pain, they will have a lot of inflammation, and it's associated with neutrophils activation and neutrophil degranulation. Okay, so then what's happened over time? Well, over time, people who resolve pain, is the yellow here, they will downregulate this inflammation. So it's going up and it's going down, and it's kind of dramatic changes. which happen in the blood of the people who resolve it, and it's not happening in the persisters. But in this moment, we only had samples at acute pain and and 3 months later. We didn't know, is it those who in acute, they always have a high inflammatory response, or they actually upregulated? I mean, logically will be they upregulated, but we didn't have data on this. So, but then we had another cohort, which we also was thinking, okay, we can replicate phenomena. We was looking for low back pain, but we didn't have one, but we had one in TMD, which is a musculoskeletal pain in the face. But in TMD samples, we had, in TMD cohort, we have healthy control, acute, then persistent in 6 months and chronic. And in this case, we did see that really inflammatory response is in resolvers, what hugely upregulated, and neutrophil activation, neutrophil degranulation, macrophage activation. It's always upregulated in comparison with control, but it was actually even low but in the T1, it was below level of control. So meaning they undershoot, right? So the resolvers, they go very up, and then they really trigger anti-inflammatory response, resolution. And already in 3 months, they have anti-inflammatory state, so they undershoot. And no, in persisters, even at T0, they have kind of below control and continue to be like this, not very prominent, but still didn't trigger inflammatory response, which is at that moment. Okay, so at that moment it was a little bit strange, because we know that chronic pain patients have low-grade inflammation. This has been documented many times in many diseases. So how come? Yes, but if we take a chronic TMD is when people have 10 years of pain. Now the inflammatory response is up. Not very much, but up. So basically this allows us to create this model when in acute to chronic transition, what we need is acute inflammation, which trigger resolution, tissue repair, and anti-inflammatory response. So in those who cannot do this, those people, their inflammatory response continue very slowly to rise and continue and just never resolve. Continue like this low-grade inflammation. So again, I wanna stress that so far, I just been in this meeting in Bergamo in the master class, and I give a similar talk. And one professor told me, I thought about this and said, well, what you suggest doesn't contradict to anything we know. And yeah, I thought it's very important it's important to stress. It's true. It really doesn't contradict. We know that chronic pain patient has low-grade inflammation. They do, and they cannot resolve it. So what the piece which we didn't know is that acute inflammation is necessary for resolution. This seems to be at least helpful. So, but at this moment, this was just the hypothesis. I mean, it's still hypothesis in many ways. So I count on you guys to be engaged and create, new results which confirm or not this result. But at this moment, I talked to Dr. Jeff Mogil, who is a very also a well-known, celebrated, recognized professor in pain in McGill, and he's an animal behavior person and pharmacologist. So we're having this lunch, and I told Jeff about this result, and Jeff said, cannot be, because we know that if we give anti-inflammatory to mice, they have analgesia. And then Jeff said, wait a minute, how long we watch the mice? We don't. We always look at the immediate effect. Let's do this experiment. It's very easy. We will watch the mice. Let's give them dexamethasone. Why dexamethasone? Because corticosteroid will inhibit everything, not only inflammation, just to make sure that we took care about all immune activity. And this is what Lucas, his postdoc, this is what Lucas did. So he took several models. So we started with very simple model of CFA, which is some people consider chronic inflammatory, some acute inflammatory, but it's an inflammatory pain model. And then we did few others, and basically Lucas inhibit or give also mice injection of dexamethasone for 6 days. Only these 6 days in the acute stage. And then he measured the sensitivity to 1 Fry, sensitivity to noxious stimuli to touch in mice. And this is what we saw. So acutely, the mice who has received dexamethasone, and this is this black dot, you see they show analgesia. By the way, yeah, if you never used to watch this mice pain kind of graph, here is the withdrawal threshold, meaning the higher the number, the less pain behavior people, mice show. And so the low, the more pain they show. So first CFA, okay, mice open circle, mice show pain behavior, but mice receiving dexamethasone, they're very statistically analgesic. But if we continue to watch mice, you see that there's this cross. And then mice continue to have pain and continue to have pain with dexamethasone. Mice without dexamethasone, they resolve pain in about 3 weeks. Mice with dexamethasone only resolve pain by day 140. So it's 3 months, more than 3 months, they show pain behavior. And both of these effects are very statistically significant, both acute analgesia and long-term hyperalgesia. So then the same effect we saw. Sorry, computer is thinking. I think it was too much. It's frozen.
Let's see. Oh, it's actually frozen. Yeah. Wait.
Yeah, so this happened to many people at this moment, who was looking at this graph. Again, it doesn't contradict anything we know. So we know, it's just the way we measure analgesia, the way we measure the test drugs in mouse model, it's always immediate analgesia. And we also think that, and it should be linear, right? Like the more good things should be good things, and the more bad things should be bad. Yeah, but it's not.
Yes, we will have to close it. Sometimes it happens. Not the best computer out there we have here. Sorry. Let's reopen. That was slide number 11. Right. And number 11, presentation. That's 7. Here it is.
Yeah.
This one. Okay. We're back.
Okay, okay. So then, then we did the same. We did the same experiments in different animal model. We did it in CFA model, not as dramatic as CCI, it's neuropathic pain model, not as dramatic as CFA, but we saw very significant prolongation of pain behavior. And also in the model of neurogrowth factor induced pain, we saw very significant prolongation in pain. Then the same, we saw it even more significant than CFA is the so-called poincisional model. It's the model of postoperative pain, right? So you do incision in the paw of the mice, and approximately the same, approximately 3 weeks, and the mice without incision, they completely heal, nothing left. But if you just treat them with dexamethasone in this sensitive time, they develop this very, very long-lasting hyperalgesia for over 3 months. And there is no difference in males and females. Both males and females do this. And it's also true for the so-called cryotherapy. An idea again was cryotherapy. So we use it all the time in the case of acute injury. So both application of ice for 60 minutes for 3 days or 30 minutes for 3 days in the mice in the CFA model. So this is sport injury model. Again, see prolonged pain for a very long time. So next question was, is it really inflammation we cannot inhibit, or it's pain we should not inhibit, right? So what is important component? Because when we give dexamethasone, it's both. So in this case, we compare drugs, different analgesic of different category, diclofenac, which is NSAID, a non-steroid anti-inflammatory, gabapentin, sodium channel blocker, and morphine. So it's all happen in the CFA model. And you see the long-lasting pain. This time we didn't, torture mice for too long because the separation was very, very clear. You can see here duration. It's only diclofenac. It's only NSAID which create this long-lasting pain. So we do need inflammation to resolve pain. So then in our pathway analysis, the neutrophils always was on the top, right? So, okay, the conclusion was probably inflammatory response driven by neutrophils. So does neutrophil really help? So what we did is we inhibit neutrophils by antibody against neutrophils instead of dexamethasone. We didn't give mice dexamethasone in this case. We just give them antibody against neutrophil, and it was the same effect as dexamethasone. So really, you see kind of almost the same switch, right? So neutrophils is needed for mice to resolve pain. And so the last in this kind of category, we also said opposite. What if we give mice dexamethasone, but we supplement them with neutrophil from naive mice? Will it reconstruct non-pain state? And the answer was yes. And we asked the same question for this protein, which called S100 protein, S100A8 and S100A9. So it's 2 protein, which is the major in the neutrophil. In fact, the content on the neutrophils up to 40% of these 2 protein. And it's a strong kind of signaling molecule. So you see the black line here is what's happening if we give mice dexamethasone, but if we give them neutrophils or either of S100 protein, they immediately resolve pain. So it's really neutrophils, at least neutrophils initiate. Right now I'm thinking, well, macrophage, we didn't check because neutrophils come first. And when they done, which they kind of working efficiently in a few days, they call macrophages. So I think macrophages will be maybe as important. It's just neutrophils come first. So then the next question, everybody asked me, wait a minute. So you said we cannot use NSAID. So we cannot use opioids anymore. And now we cannot use NSAID. So then what can we use? So we start to kind of think about this. If the whole idea that we should upregulate this adaptive inflammatory response, how it can be done? It can be done. We did a few experiments. So first, Mohammed, our research associate in the group, he came to me and he said, I just published paper that some prostaglandin agonists actually lead to upregulation of S100 protein. So how about we will treat mice? And this is already, this prostaglandin, which exist in the clinical practice for other indication. So what if we will use them? And so he did this gavage. So it was paresa for mice. And yes, immediately, you see, he gave prostaglandin for just 2 days to mice. And all this there are many of this category, but he tested Veraprost, Misoprostol, and Treprostinil, because this 3 has a different kind of range of the prostaglandin receptor activation. And all 3 of them, so look at the yellow, green line in all cases, all of them lead to resolution of this our dexamethasone-induced state of pain. Okay, so one more suggestion. We thought, how about something massive? Yeah, something like autologous condition serum, or something which upregulate our body response. Acupuncture, enriches plasma enrich plasma, platelets enrich plasma. Yeah, something which have this non-specific massive upregulation. So we use ACS, which autologous condition serum, which is just you take blood, allowed to incubate, spin, collect serum, give to patient. And so why again we thought of this? Because ACS was shown to have lipids and exosome and growth factor in inflammatory and anti-inflammatory molecules, and people usually who study this concentrate on anti-inflammatory molecules. But what we see, anti-inflammatory molecules are regulated together with pro-inflammatory molecules. It's a kind of massive response. In fact, we look at the ACS, the S100 protein increase, almost 5,000-fold in the ACS, 5,000-fold. Pro-inflammatory interleukin 8, increase over 2,000-fold. Okay, what will happen? Okay, Anahita, my graduate student, so she used our incisional PO model. It's chronic post-surgical pain, and the mice who treated with dexamethasone, again, they have this pain forever. But if she just give 2 injections, maybe it's 1 injection is okay, I don't know, but we give 2 injections of ACS kind of just after surgery, when supposedly patients are still in in the hospital, and they immediately resolve pain, or they will not develop chronic pain. Right, they did it again.
Might not be very. Oh, okay. Oh.
There's life. Okay. Okay, great. Maybe not. Oh, wait, okay. Yes. Okay, yes. So here's, we come to conclusion of the first part, which is acute to chronic transition. And so basically what we saw is bad for our patient, and what we saw is good for our patient. And I'm talking about situation of acute to chronic transition. It can be surgery, it can be trauma, it can be acute low back pain, right? So corticosteroid, NSAID, cryotherapy is bad. So they may promote this acute to chronic pain transition after pain or trauma. And so very important that what I say may is there is also, we shouldn't think that we as a clinician create this pain state like solely. No, there is also intrinsic ability of people to, to upregulate the inflammatory response. In fact, in our original cohort from Dr. Massimo Allegri, basically everybody was having NSAID because it's a standard of care. But some people still, had enough upregulation and some didn't. So, but as we see in the animal model, we kind of can help people to resolve, or we can be on their way to resolve. And so, but what was good, what neutrophil was good, S100 protein was good, a prostaglandin agonist was good, autologous condition serum was good. And like what we hypothesized that any controlled pro-inflammatory methods will be good. Again, acupuncture is one, maybe physical therapy is doing, not maybe it's not, physical therapy create control local inflammation. So this may prevent acute to chronic pain transition. So maybe we can think about how to help our patient after surgery, right? So here's the conclusion number one, that there is an active adaptive component in the immune system in acute to chronic pain transition. And again, we already saw many cohorts. It's very, very basic. It's always happen. So in inhibiting this immediate active adaptable responses in human with physical injury, we lead to pain chronification. So which means that in our drug developing, protecting against chronic pain, because I think we need to differentiate in our mind, right? Do we need immediate analgesia, or we worrying about chronic pain? It's not the same thing. So that's why we need to think about developing against chronic pain. The animal model should target pain resolution. We should upregulate pain resolution. And also we may target activation of immune system, not inhibition of something. And finally, that we may need to stay away from inhibition of acute response. And it's maybe we need to kind of, not maybe, I think we should study this and be trying to do this kind of when, because it's all going up, going down, but at least we need to be aware of this. But the next question is, then how about chronic pain resolution? So we talk about acute prevention, right? How about resolution? Because, for example, we asked this question and Anahita said, well, it's very easy. So let's give, for example, ACS. I will give you two examples, but this is just the ACS. In the moment when it's already day 30, when the mice who treated with dexamethasone, they have long-lasting pain. The mice who don't get dexamethasone then resolve long time ago. So this is kind of, and this is po-incisional model. So this is model of chronic, when the 1 month when people after surgery, they report pain. And so majority of the people don't, but if people report pain 30 days after surgery, it's a very strong indication this person will also report pain 3 months and 6 months after. So this is 1 month. Okay, if in this moment again we give mice ACS, the mice immediately resolve the state. So mice needed just a little kick at this moment too. It's not only prevention, it's also resolution. And the same true, I figure out, yes, I figure out what's the problem. If I try to change slide okay, yeah, but anyway. Yeah, so the same true actually the Lucas did for day 30. So he just make mice, all mice was developing a chronic pain, day 20, the mice, it's post-surgical model or CFA, no, CFA. The mice who didn't get dexamethasone, they already all resolve. And then, yeah, and they, yeah, so this is kind of what's happening, right? So mice was having this long-lasting pain. But if he give them already in day 28 neutrophil or S100 protein, they immediately resolve pain. Again, this just a little bit of kick with the inflammatory signaling. So then we ask, Is this can be then indication, the ability of chronic pain patient to upregulate their inflammatory response? Can it be also indication that they can resolve and can we help them then to resolve, right? So what we did is we already have actually cohort, but idea was, yes, so can we give the inflammatory stimuli to neutrophils and we give them a low LPS. So LPS is a bacterial wall. So how well they respond to LPS? This was the whole idea of this exercise. So the cohort has been collected by Carol Milotter when she was a postdoc, and it was sitting in the freezer when Sahel, my graduate student, said, okay, I want to analyze the RNA of this fibromyalgia and healthy control, and also the activated neutrophils. So she did the full transcriptomics of these people, analyzed pathway and the gene expression, but we also look at long-term outcome because this cohort was, 5 years old. We wanted to see if people resolve pain or not. Oh, do they get better? Fibromyalgia, we usually believe they don't get better, but they do. So we deliver them questionnaire, we call each of the patient and we ask, okay, with this questionnaire, how did you do? And we found people who get better and who didn't get better. So and how the transcriptomics is different. So first, the result is a baseline. At the baseline, no activation of LPS, nothing. What is the difference between fibromyalgia and control? And this is again famous volcano plot. Each dot is a gene. So a lot of these blue dots, which is statistically significant, upregulation in fibromyalgia. Many genes upregulated in fibromyalgia patient. And if you do pathway analysis, it's all about inflammation. So number one, the most significant is inflammatory response. Yes, as we knew, fibromyalgia patients have low-grade inflammation. No question about this. Yes, so it's a low-grade inflammation what they have. But then what happens when you stimulate the neutrophils? And this is where we see also big difference. So this is control. You see enormous amount of the blue dot all above significance, basically all genes upregulated. Neutrophils, the CLPS, they're like going crazy. And it's a thousands and thousands, 10,000 fold upregulation. What's happening in fibromyalgia cases? Yeah, they are upregulated, but you can see not that much. It's very blunted. And this is logarithm of p-value. It's very significant difference. And it's very significant also the test is significant, like really not in a specific gene, but like all together. I mean, each specific too, but mostly all together. But it's also interesting that if you do the pathway analysis, then controls, then they show many significant pathway, majority of them response to LPS, inflammation. So the normal neutrophils, they react as they should react. They respond to inflammatory stimuli. But fibromyalgia patient, even they have this many statistical genes, there was only like 9 statistically significant pathway, and it's mostly metabolic something. So it's look two things. The first, the response is very blunted, and it's dysregulated. Even those they have upregulated, they're not converging very well in inflammatory response. So they confused, they exhausted, they're not well. So next, what Sahel did is interaction tumor analysis. And interaction tumor analysis is exactly you asking, okay, looks like, think about this, that fibromyalgia patients, they had upregulation of inflammatory response in the baseline, but then they cannot upregulate inflammatory response based on the stimuli. So it was interaction. It's like this, right? So this is what the statistician kind of, this is the strongest interaction. And so when Sahel did pass interaction term analysis, yes, many genes, yeah, exactly this. And I will even show you exactly how it looks like. All classic inflammatory gene, they in the baseline, they a little bit higher, because every time people tell me, oh, maybe fibromyalgia patient cannot upregulate, the pro-inflammatory cytokine because they already maxed up. No, they're not maxed up at all. So even all this low-grade inflammation is really, really little before stimulation. It's statistically significant, but it's quite little. But it's just after stimulation by LPS, they cannot upregulate it as well. So the next step is, remember I was telling you, improver persisters. So we differentiate the people, fibromyalgia subject in these two groups. So has been collected by our clinical study coordinator, Francesca Montagna. And so at the baseline, remember this, I show you, okay, fibromyalgia patient have upregulated pro-inflammatory state, but not improvers. Those who will improve 5 years before they improve, they don't show this low-grade inflammation, or very, very little, non-statistically significant. And those who will be persistent 5 years later, they have this very, even more significant low-grade inflammation they control. So this control is driven by persistence. So next is how they respond to LPS. Okay, this is original like control, like huge volcano plot. But improver is, yeah, they blunted response, but they kind of get in there. When the persisters are those who really cannot upregulate their responses. So what's interesting also, if we look at all the psychological questionnaire or even fibromyalgia survey or anxiety and back depression, none of them were different between persister and improver and the baseline. So they look in terms of their psychological output, they was looking, very, very similar. It's the neutrophil who were different. So next is, okay, let's see if we can do it. Yeah, so we ask if neutrophil create the specific state in our body. And so, of course, we care about sensory neuron in pain. So, okay, so this culture of sensory neurons, and we treat this sensory neuron culture with plasma from fibromyalgia patients. And you see the signaling, it's very stimulating for the sensory neuron. So the next, yes, so we compare, so this is just example of how we measure calcium responses, and Skip and Sarah in the College London was doing this experiment, helping us because we would never do this ourselves. And this is where we see big difference between control, improver, and persisters. You see that it's a persister who really stimulates sensory neuron, and improvers and somewhere in between. And the range of the activated sensory neuron, it was really increasing as the persistence of the disease was increasing. So the next, Sahel said, well, yeah, I know that Luda, you always say, oh, it's just everything is involved, but it should be, or it could be, or it will be nice to find the master switch somewhere up. I said, well, if you want a master switch, I mean, there are few, but transcription factor is one of them. She said, yeah, easily, I can do transcription factor analysis. So she did transcription factor analysis, and the only significant component was between cases in control or in an interactive model. It's all different components of NF-kappa-B transcription factor, which is not, too surprising, because NF-kappa-B is the inflammatory regulator. It's a master inflammatory regulator. But again, maybe what surprised was is the direction. Because when we look at the pathway analysis in an FKPB cases, there was the one who was has a very significant negative regulation of an FKPB. So in other words, the NF-κB was inhibited in neutrophils. So not activated. You would think, okay, so they have low-grade inflammation, it should be activated. It was inhibited, and even more in persister in comparison with improvers. But I think this is what's happened. The cells see that there is low-grade inflammation, and cells try to deal with it. How cells deal with it is inhibit NF-κB. But because cell inhibit NF-κB, it cannot upregulate proper inflammatory response. And it's trapped in this low-grade inflammation further and further. The more it's going, the further it will be trapped. So we just one more kind of series of experiments I will show you because we wanted, because it means that in fibromyalgia patient and chronic pain patient, we need to actually upregulate, we need to stimulate NF-κB, which is opposite to what we usually think, right? We usually think we need to inhibit NF-κB. There is a lot of clinically available NF-κB inhibitors, not only chronic pain, in all neurodegenerative disease. It always has been tested. But okay, we said, how about we we stimulate NF-κB? There are a few. We found 2 clinically available upregulator. But we kind of, what model? We discussed, like, I don't believe any model of fibromyalgia. So then we said, how about we do plasma transfer? We took plasma because we knew already that plasma in fibromyalgia patient, there's something there, super significant. So if we give plasma to mice, human plasma of controls, nothing happens. If we give them plasma from improvers, they actually start to show some pain behavior. If we give mice plasma from persister, they show this huge hyperalgesia for about 3 weeks. So just the plasma there, right, contain this biologically active molecules. So, but this now it's became a good model for us to test the drug. And this is what we did because, okay, we took briastatin. Briastatin is clinically available activator of NF-κB, purified from this nice urine plant. So if this is the control, if we give briastatin to just mice who treated with control, mice actually develop hyperalgesia for, less than 2 weeks. So it's hyperalgesic for mice. But if we give briastatin to mice with the who treated with the persistent plasma, they immediately become analgesic. So they immediately resolve this hyperalgesia. So with this, I want to just conclude that I remind you, so with the chronic pain and the baseline fibromyalgia case show activated inflammatory phenotype driven by persisters, but they cannot be activated very well. Although in the baseline also psychologically, we don't see big difference. We don't see any significant difference between those who will improve and not. And finally, that NF-κB seems to be this most important transcriptional factor in neutrophil contribution. So maybe proper stimulation, stimulatory response of neutrophil can underlie, first of all, natural resolution of the chronic pain patient. In other words, I think that people get better because they have this ability to get better. So finally, something will happen to them. They will do exercise, they will do acupuncture, they will do meditation, develop positive mood. So finally, they will resolve it. But of course, we can help them. And so maybe they don't need 5 years. Maybe we can help them this way. And the way how we can help them, we may help them maybe with the drug I like brastatin, something which upregulates, stimulate the immune response and inflammatory response and stimulate NF-kappa B. So in overall conclusion, and I saw this with other, so I told you about neutrophils, but I saw it with other cells. Chronic pain patients, neutrophils, exhausted, confused, non-responsive, and we need to restore their health. So how to do this? I'm thinking of this, but I would like to have a discussion about this. And thank you. I would like to introduce our program in pain genetic in McGill, which is not all people who work for me. We have a program with professors there, all working on human pain genetics. And I would like to say thank you to all funding agency and to you for coming. Thank you very much.
Yes, please. Okay.
Thank you for possible that the response is in some way affected by the dose of the drug we are prescribing.
Yes, I'm sure it is. Yeah, I think it's a combination of our intrinsic ability or each patient intrinsic ability to heal, but of course it's hugely affected by the drugs. And that's why I think that right now our standard of care is after surgery to give NSAIDs, but maybe we should be rethinking and giving something else, or in addition to NSAIDs, give something which help our immune system to stimulate the immune responses. Yeah, so we did, this is an interesting question. So we did a lot of genetic analysis because transcriptomics has become affordable only maybe last 7 years ago, but GWAS was affordable before. So we did a lot of GWAS, we did a lot of also analysis of UK Biobank and other big cohorts. So if you do partition heritability, for example, in other words, you take this whole genetic data and you map it to tissue-specific gene expression. So what's interestingly, we don't see mostly immune system. We see brain, and some immune system. We do have some immune system. So, I mean, I'm convinced that the gene-environment interaction, this brain immune interaction. So what I don't know who is come first, or maybe the question doesn't have an answer because maybe it doesn't matter where patients get into this loop. Because we see both, but in the partition heritability we mostly see brain. I have at least two explanations. First is because the contribution of immune system is bimodal, we will probably not see it in GWAS. Because if you have, for example, a genetic variant which code for high in FKPB expression, is it good or not? It depends, because it will protect you from acute to chronic transition. But if you maybe already in the chronic, then you continue to have this high. So this one explanation, because it's not linear. And the second, maybe immune system is not the most important. Maybe brain is more important. Yeah. Well, if I understood question correctly, if inefficient inflammatory response at the acute stage, I would, this is my, message. Yes, it can lead to the chronic inflammation and autoimmune response. I can imagine the inflammatory response can be also too strong. But maybe this is what we're dealing with. Can it be too strong? Can it be too little? Right? Should be exactly right. I hope I answered the question. Well, thank you for coming. Thank you very much. Yes. Thank you. Thank you.
I'm fascinated by this question that chronic inflammation is at the beginning of, I think, a lot of pathology.
Yes.
But at the moment, also in pediatric life, we also inflammation.
Okay.
This is very dramatic condition probably you way in which you live probably. I don't
Yeah,
know.
because we always need to inhibit inflammation.
Yes, I don't know, but in my mind, I that there is an mental condition that help doesn't help us to resolve the problem.
Exactly.
And resolve the problem. We don't have more habit. Our system, immunological system respond probably in a different way. Yes. Today, with respect to the past, I don't know. I ask you this is
I think we definitely, we tempering this our immune responses, and we don't usually look at the long-term outcome. We don't know how our treatment affects long-term outcome. This data suggests that it's bad.
yeah.
It's bad. So we need inflammatory response, because again, it's not an inflammatory response, then it's trigger growth factor release. Like in the trauma, for example, in the bone healing, it's already they try to vote inside because it's very clear that it's inhibit bone restoration and bone healing. Right? So I think it's not only this, many. So in fact, in the chronic pain case, we know that it seems to be more and more people from pain. But the argument is, oh, before we just didn't measure this properly.
Yeah.
Well, we don't know.
We don't know.
We don't know. But interesting about Alzheimer, for example, people also say, oh, before we didn't lead to our Alzheimer, and you have didn't properly measure. But Alzheimer originally has been described as a peculiar rare disease.
Yes.
And interestingly, I've just been in Louvre last year, and I looked at the room of the great French scientists, and I looked specifically how long do they live. Actually, they live 80, easily half of them. Yes, so it wasn't that really. I mean, I understand that more people die early, But it's not all of them. Okay, maybe Cremonius, died. So it's
Thank you, thank you. Qui possiamo fare tutto.
Aspetta però Alessandro, tanto lo riporto io perché ho notato che ci sono stati diversi problemi. Innanzitutto il microfono non sembra. This.